back everybody to learn medicine by Dr. Nikita. Today we have brought another interesting topic and you must have learned and read a lot about receptors and how is the insulin working in that and what is the role of insulin, what are the pathways involved and what is happening inside the cell, how is the glucose being reuptaken, what is happening to glucogenesis, glycogenolysis, all those topics are interlinked by the things happening inside the cell. That is the role of secondary messengers, the role of phosphorylation cascades. You must have heard of the term MAPK. So all the things I'll be addressing today. So our topic for today is understanding the mechanism of insulin and what is the role of receptors associated with it. So first of all, insulin is bound to the insulin receptors which are present in many places. There are many insulin receptors on maybe cell 1, I'm just calling it. So there are so many cells which might be having insulin receptors. But the insulin receptors are dense in the adipose tissues. They are dense in liver cells. They are dense in muscle tissues. So there are a lot of places where the insulin receptors are dense. Insulin receptors are tyrosine, are belonging to the tyrosine receptor kinase family, and they are heterotetrameric. So they have 2 alpha subunits, or we can say 2 alpha chains and 2 beta subunits or 2 beta chains. So let's again come back to where is a brief idea about from where the insulin was being released. I have discussed a lot about insulin in my previous lectures. You can go check that out. Just a brief revision of that. So here is the pancreas, which is responsible for the pancreas has beta cells responsible for the release of insulin, right? And this insulin, with the help of portal vein, is entering the liver. But when it enters the liver, when this insulin released from the beta cell of pancreas, I'm saying this is pancreas, and pancreas is having beta cells which synthesizes insulin. This insulin is entering with the help of portal vein into the liver. Here there is IDE present. Okay, IDE is an enzyme responsible for degradation of insulin. So IDE, that is insulin degrading enzyme. Insulin degrading enzyme. Insulin degrading enzyme is responsible for degradation. As the word in itself says, degrading, that is degradation or breaking down. So this enzyme is degrading what? Insulin. Degrading enzyme. So this enzyme is degrading the insulin. So insulin which is entering the liver with the help of portal vein is degrading more than half of its volume of what it has been synthesized in the pancreas. And here less than half of the volume of insulin is now remained, which is flowing into all these cells respectively to its receptors, and these receptors are although present in numerous places, but they are mostly densely found in adipose, liver and the muscle cells. So now what is the game happening here? When this insulin reaches these cells, what is happening inside? That is what we have to discuss today. So I hope you had a brief idea about this. I'll again draw a clearer flow chart with minimum pointers to be collected here. That if I say pancreas is responsible for the beta cell of pancreas are responsible for synthesizing of insulin release, right? And this insulin with the help of portal vein is entering where? The liver. So liver is having IDE enzyme called as insulin degrading enzyme, which is degrading more than half of the volume of insulin. So now less than half of the volume is, or more than half, less or more half of the volume of insulin is being remained depending upon the release of IDE, and that insulin is now being circulated to all the body parts. And we all know that what is the role of insulin? Insulin is responsible for increasing the uptake of glucose from the blood cells. Ultimately, it is responsible for suppressing the glucose concentration in the bloodstream, and it converts the glucose into glycogen. That is how it is stored into the various places, or it is responsible for storing it as glycogen, increasing the glucose uptake and substantially decreasing the concentration of glucose in the blood. So this is a brief idea. After explaining all the topics, what I have introduced new is I'll be listing out that in the form of a flowchart. This will help you to draw the notes or write down the notes and reciprocate the same in your examination. I think that will be helpful. You can drop in more suggestions if you want different kind of explanations, right? So moving on to the next topic, I hope this is clear. Again, repeating the same thing, that pancreas is having alpha cell, beta cell, delta cell. We have discussed in the previous lectures, where the beta cells are responsible for synthesis of insulin. This insulin is carried from the portal vein to the liver, and here liver is having IDE, that is insulin degrading enzyme, which degrades more than half of the insulin, and only what is left of it is being circulated to other body parts. Now this insulin which is being circulated to other body parts, this insulin now acts as a ligand. I hope all of us know what is a ligand. Ligand, whenever it binds to its receptor, it creates some kind of a response in it. So I already told you that insulin receptors are heterotetrameric. So now let's see how it looks like. We have two alpha subunits and we have, I think you must have seen this kind of a structure in various books, in KD Tripathi also. So this is the beta subunit and this is the alpha subunit. So the interesting part about the insulin receptor is that first it belongs to the tyrosine kinase domain. Tyrosine kinase receptor domain. The insulin receptor belongs to the tyrosine kinase receptor domain. So what do we understand from this term? First of all, there is one enzyme here mentioned called as tyrosine kinase. So tyrosine kinase has a major role to play here. Okay. So this belongs to that. The insulin receptor belongs to the tyrosine kinase receptor domain. So it has two subunits, or we can say it has two chains which are present there, alpha and the beta. So now this has a unique identity that half of the receptor is present outside the cell. This is the cell membrane. Okay, this is the cell membrane. So half of it can be seen outside the cell membrane and half of it lies beneath it. So what part is lying outside of the cell membrane? This is called as the alpha subunit or the alpha chain. Half of it is lying beneath it, that is the beta subunit or the beta chain. So once this insulin molecule, this insulin released from the pancreas, from the beta cell, through the portal vein, through the liver, when it is being released to the body parts, this insulin, I am just rubbing this off so that you get a clear idea. I am rubbing this off also because we have a gist here now. So this insulin is now finding its receptors and binding to its subunits, right? And when this insulin binds to its subunit, it activates the alpha subunit, right? When it activates the alpha subunit, this alpha subunit in turn activates the beta subunit, and beta subunit is having a tyrosine kinase activation. As I already told you, it belongs to the tyrosine kinase receptor domain. The beta chains, they trigger their tyrosine kinase activity, and this tyrosine kinase activity is responsible for phosphorylation of the tyrosine residues. We already have tyrosine residues present here, and these tyrosine residues are now phosphorylated. I am writing PO4 in front of that, so you know that they are now phosphorylated. So these tyrosine residues which are already present there, but they could not be phosphorylated. Why they could not be phosphorylated? Because initially the tyrosine kinase was not active. How it became active? after the beta chains activated the tyrosine kinase activity. How is the beta chain being active? When the alpha subunit are activating the beta chain. How is the alpha subunit activating the beta chain? When insulin, which is having the ligand binding site, when alpha subunit, which is having the ligand binding site, it binds with the ligand molecule, that is the insulin. So this is the ligand binding site which is present on the alpha subunit. Insulin binds to the alpha subunit, triggers the beta subunit or the beta chain. That beta chain triggers the tyrosine kinase activity. Kinase means that we are talking about an enzyme. So the tyrosine kinase in turns phosphorylates the tyrosine residues. So now this is the cell membrane. Okay. I hope all of us know how the cell membrane looks like. It is a phosphobiolipid layer. So inside this, okay, so here is the stream of IRS. So IRS stands for the insulin receptor substrates. What is IRS? Insulin receptor substrate. So IRS are already present there, but they are only attracted when the tyrosine residues are phosphorylated. So the IRS are now binding to the binding here, and they are also becoming phosphorylated. What kind of IRS are there? There are two types, IRS1 and IRS2. So the insulin receptor substrates are now phosphorylated, and now they trigger two pathways. Okay. I am just mentioning the two types. It is not crossing the cell membrane. I am just explaining the two types. Everything is happening inside this cell membrane. Everything is happening inside the cell membrane. So the IRS is triggering two pathways. One is the phosphorylation cascade. Phosphorylation cascade. And the other one is PIP3. Okay. And the other one is the PI3K. The other one is the PI3K. We can also call it PIP3. That ultimately PI3K will lead to the formation of P2 and PIP3. But it is called as the PI3K, and this is called as the phosphorylation cascade. So the phosphorylation cascade is responsible for two things. This causes the phosphorylation and the dephosphorylation, that is the activation and deactivation of the enzyme. So everything what was happening here can be triggered and suppressed depending upon the phosphorylation cascade, right? Phosphorylation cascade in turn it leads to the activation of MAPK pathway. Okay, this leads to the activation of MAPK pathway, and PI3K is also called as the phosphoinositide 3 kinase. Phosphoinositide 3 kinase. So these are the two pathways now we will be learning about. I'll quickly have a brief discussion or revision of what we learned earlier, like now in the lecture, and then we'll be majorly focusing on these two pathways, the PI3K pathway and the MAPK pathway. So let's again go back to this thing. Pancreas were responsible for, the beta cells of pancreas were responsible for the synthesis and release of insulin, which through the portal vein enters the liver with the help of IDE, that is the enzyme, insulin degradation enzyme responsible for degradation of more than half of the insulin. Whatever is left of the insulin is now being transported to the insulin receptor rich areas, that is the adipose tissue, the liver and the muscles. The insulin, which is the ligand here, binds to the alpha subunit, which is the ligand, which has the ligand binding site and is extracellularly, that is present out of the cell membrane. So it binds here. This triggers the beta subunit, which is lying inside the cell membrane, and here the beta chains are triggering their tyrosine kinase activity. This tyrosine kinase activity leads to the phosphorylation of the tyrosine residues. This phosphorylation attracts the IRS, and this IRS1 and IRS2 are responsible for the insulin receptor substrates. They get phosphorylated and they trigger two pathways, the PI3K pathway or the phosphorylation cascade, which is triggering the MAPK pathway, mitogen activated protein kinase. So I'm just rubbing everything off, and now we learn in detail about the PI3K pathway and the MAPK pathway. Okay. So from here we had two pathways clearly, the MAPK pathway or the MAPK signaling, and the other one is PI3K, phosphoinositide 3 kinase. Phosphoinositide 3 kinase. So the PI3K, it leads to the phosphorylation. This PI3K, it leads to the phosphorylation and forms PIP2, which stands for phosphoinositide 4,5 bisphosphate. Phosphoinositide 4,5 bisphosphate. Let me just finalize it. Inositol. Yes, inositol. Phosphatidyl inositol 4,5 bisphosphate. This was phosphoinositide 3 kinase. So PI3K leads to the formation of PIP2, and PIP2 with this phosphorylates to form PIP3, which stands for phosphoinositol 3,4,5 trisphosphate. This is trisphosphate, or we can call it 3,4,5 trisphosphate, phosphoinositol 3,4,5 trisphosphate. This PIP3, now it leads to the formation of PDK1, which is also called as phosphoinositide dependent kinase. This is called as phosphoinositide dependent kinase. This PDK1, it leads to phosphorylation and formation of AKT, also called as PKB. This is protein kinase B. So now you understand the whole process. PI3K, it phosphorylates to form PIP2. This phosphorylates PIP3, and this phosphorylates the activation of PDK1. This phosphorylates the activation of AKT, and which is also called as the protein kinase. Now this AKT is responsible for all the cellular changes happening here. And how? Because the AKT is responsible for transportation of the glut to where? This glut is being transported to the cellular membrane. So now this cellular membrane will have a glut. What is glut? Glucose transporter. So any glucose which is present in the bloodstream, this is glucose, can be easily uptaken back, right? Just because insulin binded and triggered all these processes, which ultimately leaded to the formation of the glut vesicles being pushed back to the cellular membrane so that they can intake the glucose present in the bloodstream. I hope you get the entire gist of the mechanism. So whatever excess of glucose was present in the bloodstream, that is uptaken, that is absorbed into the liver cells, the muscles, the adipose, with the help of insulin, because insulin binded to alpha subunit, triggered the beta subunit, it triggered the phosphorylation cascade, which triggered these two pathways, and ultimately the glut transporters or the glut vesicles were pushed to the cellular membrane to capture, to grasp, to collect the excess of glucose present in the bloodstream, right? So this is this. Now we will be focusing on the MAPK pathway. So we know how the glucose is being taken. This is the bloodstream flowing here. Okay, I'm just again mentioning, this is glucose freely flowing in the bloodstream. This is the bloodstream, and this glucose is now absorbed and is now entering the cellular membrane, crossing the cellular membrane, entering the cell, right? So this cell could be a cell from the liver, this cell could be a cell from the muscle, could be a cell from the adipose tissue. Anyhow, whatever the cell is, the blood is now free or is now having a decreased quantity of glucose. That was the exact role of insulin. Insulin was responsible for decreasing the glucose concentration. So that glucose is either going into the liver cells, either going into the muscle cells, or going into the adipose. Right? In all the three places, in muscles, it is utilized as a source of energy. In the fat, in the adipose, it is stored as glycogen, and similarly, it goes for the liver also. Right? And ultimately, the entering of the glucose in the liver cells will also be leading to suppression in the glycogenolysis. Right? So that also we'll be talking about in next lectures. So I hope this is clear to you. You just have to memorize the names here, that PI3K will be phosphorylating, the PIP2 phosphorylating into PIP3, PDK1, AKT, and finally this is responsible for pushing the blood transporters to the cell membrane for catching the glucose which is freely flowing in the bloodstream, capturing it, and this is how it is storing it inside the respective cell, whatever cell this is. Could be a liver cell, could be an adipose cell, could be a muscle cell. So I'm just rubbing this off so that we can discuss the MAPK pathway. Okay, now next. This has become a bit untidy. I'll just clear it off and again give you a brief idea. So this was the alpha and this was beta subunit, right? And I did told you that the glucose transporters are now active. This is the glucose uptaken inside the cell. This is the ligand binding site to which the insulin is bound. This was already phosphorylated and this led to the triggering of. Okay, now this is clear. This is what we discussed already. Okay, so now the next pathway is the MAPK. Okay, so the next pathway is the MAPK pathway. So that is also triggered. In the case of MAPK pathway, what is happening when the MAPK pathway is triggered? MAPK stands for mitogen activated protein kinase. Mitogen activated protein kinase, right? So in the MAPK pathway, the beta subunit, okay, we know what is the beta subunit and we know where is IRS. The beta subunit and the IRS, they are responsible for the formation and activation of SHC and GRB2. GRB2 stands for growth factor receptor binding protein 2. Growth factor receptor binding protein 2. So the SHC and the GRB2, which are being activated by the beta subunit and the phosphorylated IRS, they collectively synthesize and activate the phosphorylation of SOS, sons of 7 less it is called as, but let's just call it as SOS. So the SOS now is responsible, this SOS is now responsible for, I'll just make down it very clear. MAPK pathway is triggered when the beta unit and the IRS forms SHC, GRB2. This SOS is responsible for the removal of GDP. Okay, the removal of GDP from RAS. The GDP is removed by SOS. SOS removes the GDP from RAS and forms RAS GTP. Okay, the GDP is removed to GTP. And this RAS, it leads to the formation of RAF1. What is RAF1? MAPKK. Okay, this is MAPKK, that is mitogen activated protein kinase. Okay, so this MAPKK, it leads to the formation of MAPKK, also called as MEK, and this MAPK is leading to the formation of, so this MAPKK is now leading to the formation of MAPKK, right? That is mitogen activated protein kinase. This MAPKK is now leading to the formation of MAPK, that is mitogen activated protein kinase. So all of these are bounded on a scaffold protein. Okay, this is why they are closely bound together, and all these processes are triggering fastly. This is like a domino effect. This is leading to the formation of MAPK, which is leading to the formation of MAPK, and when finally MAPK is activated, it is released from this scaffold protein and now enters the nucleus. This is the cell in which it is there. If I say here is the nucleus of the cell, then MAPK, which is formed out of this entire chain reaction, this MAPK is entering the nucleus, and inside the nucleus we have what process is going on? Transcription and translation, because there are genes responsible for controlling the protein synthesis. So transcription and translation processes, I'm not writing the entire spelling, and the transcription and translation processes are being controlled or are being altered by this activation of the pathway, and this is responsible for cell proliferation and differentiation. The cell proliferation, and again I'll write the full form here if you want, proliferation and cell differentiation. So this cell proliferation and differentiation is responsible for the cell survival, for the cell growth. So all of that factors are being controlled because the glucose which is being uptaken is now responsible for cell building, cell growth. Here also, when the other pathway, that was the P13K pathway, was activated, there also the glucose was being uptaken and stored into the liver and the muscle cells and the adipose as fat, or it was utilized for the, or was exhausted by the muscles, or was stored in the liver. So all that glucose molecules which are being absorbed or being intaken have a definitive fate. Only that process is only happening when either of the pathways are activated. The pathways are activated only when the subunits are triggered. The subunits are only triggered when the alpha subunit, which lies on the extracellular, that is outside of the cell, is binding to insulin. So all of that is happening because of insulin. So I'm just bringing again a gist of all we learned today, that insulin is responsible for reuptake or for absorption or for decrease of the glucose concentration from the blood inside the liver cells or the fat cells or the adipose, wherever the receptors of insulin are higher or dense in number. All in that thing, these pathways are being activated, and we know the fate of both the pathways that we discussed. So it is like one domino effect, that ligand is binding to the receptor, and when ligand is binding to the receptor, there are secondary messengers which are being released, and they are responsible for all the cellular functions. Their cellular functions could be anything depending upon what cell we are talking about. So this is the stepwise process which is happening, and we can discuss a lot of topics related to pharmacology, pathophysiology, and topics covered in medicine, people doing masters in pharmacology, people doing masters in pharmacology, pathophysiology, and anatomy, all of that could be covered. So do follow my lectures on YouTube. We can also have a YouTube live in near future when we are covering more topics here. You can drop the topics you want me to cover in the comment section, and what I'll do is I'll link all the social media platforms so you follow me everywhere in the description below. So this was a continuation of the previous video I took, and we can similarly take up now hypoglycemics or any other topic which is required by you as per your examination or something. I'll be covering syllabus topic wise in all these lectures. So thank you. Do follow Learn Medicine by Dr. Nikita.